Okay, I think that we can start. Good afternoon, everyone. This webinar actually is the fourth webinar organized by the Aerobic Granule Working Group and EBNet, Aerobic Granule Technology and Nutrient Removal and Recovery. Today, we are very honored to invite Paul Lavender, Director Water Utilities UK and Royal HaskoningDHV to give us the presentation about aerobic granular sludge and their commercial technology. Paul is an expert in the water industry with over 24 years of experience in water and wastewater treatment, research and innovation. He is a chartered scientist, chartered environmentalist, and a fellow of the Chartered Institute of Water and Environmental Management. Today, his presentation topic is Prepare for Tomorrow, Protect the Resource Today, with the main focus on Nereda, commercial aerobic granule technology. If anyone in the audience has any questions, please type them in the chat. We will address those questions after the presentation. Paul, you can start.
Okay, thank you for the introduction, and yeah, nice to see so many people who've joined the call today. Can you hear me okay? Can you see the slides okay?
Yes, perfect. Yeah, but not a presentation mode. Yes, great.
Okay, so yeah, so I'm going to be talking a little bit about Nereda, which is our aerobic granular sludge technology. And I'm going to be talking about our, I guess, our practical experience of implementing the technology, and just give you some examples of how we've done that and some of the operating experiences that we've come across. Yeah, as was said, if you've got any questions as we go, please, yeah, just pop them in chat. I look forward to discussing those. So just a quick introduction as to who we are as a company. So Royal HaskoningDHV is an employee-owned Dutch engineering consultancy, been around for a very long time, and working globally. So we've got experience of deploying the Nereda technology in a lot of different environments. So we've got a portfolio of different technologies. Nereda I'm going to be talking specifically about Nereda today in terms of the aerobic granular sludge, but we've also got digital tools like AquaSuite and also anaerobic digestion technologies in terms of our Ephera and Helea. So I'm assuming that because everybody's joined this call, you've probably all got a fairly good working knowledge of what aerobic granular sludge is. But just, I suppose, a very, very quick kind of recap on that. So we're looking at, instead of flocks that we see in an activated sludge process, we're looking at aerobic granules. So the larger, faster settling granules. And the key advantages of that being that we've got different zones within the granule. We've got an anaerobic zone, a anoxic zone, and fully aerobic zone. So we get good biological phosphorus removal and good total nitrogen removal. So I'm aware there's various different technologies and configurations of this, but the way we deploy it is as a batch-wise process. We have multiple reactors, and all of those are operating on a sequence batch. And we have a kind of machine learning controller that then controls those batch times and the kind of recipes and how they configure. We are also working at the moment on a continuous version of this, and that's going through development at the moment in the Netherlands. So you've probably seen these before. Granules typically look a little bit like soggy cocoa puffs, or perhaps kind of a caviar or something. And we have done actually quite a lot of research on the conditions that affect the size of granules. We've also found how we can optimize the granule size and keep them at a size where we get the kind of best kind of activity. So we do see slightly different properties depending on the size of granules that we get. Something I can go into in a little bit more detail. So why do we want to look at aerobic granular sludge? Obviously, there's quite a few advantages that we want to try and exploit from this technology. We can hold a greater biomass inventory. That biomass inventory settles more quickly when activated sludge, so we can have a much, much smaller footprint. We also can use less power for a number of reasons, and I'll come on to discuss that on a slide specifically about that. And also we aim to use less chemicals. So there's a lot of consents coming in the UK, across Europe and elsewhere for phosphorus. And if we can get a good, stable biological phosphorus removal process, then that saves us having to use ferric or other coagulants to chemically remove the phosphorus. So I'm just going to show a very quick video just so you can see on a practical level the difference in settlement. Hopefully you can all see this. If you can't, if someone lets me know. So we can see in just a few seconds, these granules settle very, very rapidly. So that's granular sludge on the left, activated sludge on the right, and we can compare that in a few seconds, we get that kind of settlement compared to minutes and minutes. It speeds up now. So we see after, say, 5 minutes or so, then it's still kind of settling. So obviously that means we can design processes a lot smaller. We can allow less time because we know we don't need additional time for it to settle. We can also ensure very, very low suspended solids in the effluent. Okay, let's skip out of that. Oh, I'm not going to get stuck in YouTube, am I? Hang on. There we go. So I think Nereda is probably the most widely deployed technology using granular sludge. I suppose there's a question as to where the boundaries of granular sludge are and where there's densification processes. So there's a number of other processes out there that aim to create bigger or heavier flocks. But in terms of kind of true granulation, that's really the kind of space where we aim to be with Nereda. So we've got over 120 projects worldwide across 24 countries. We've got 74 plants in operation. And the way we control it with this machine learning controller, we're getting data back from all of those plants all of the time. So we've now got, I suppose, hundreds of years worth of operational data in total when you put it all together. And we're constantly learning, constantly evolving and tweaking and changing the actual design of the process. So it allows us to provide more kind of flexibility in terms of different ways we can implement the technology. So I'll give some examples of that. So the most obvious way of implementing it would be a new greenfield treatment site, or perhaps a brownfield site where we build it alongside an existing kind of wastewater treatment works. But we can also we can retrofit. So we can actually take existing tanks and we can turn those into a granular sludge process. The biggest challenge with that is the depth of the tank that we need. We'd say typically a minimum of 6 meters or so to make that viable. We can do it in parallel, so we can just have two, Nereda and conventional operating side by side. Or what we've done quite successfully is what we call a hybrid, where we effectively waste the granular sludge out of a Nereda plant into the activated sludge plant to the side of it. And the advantages we see of that are that we get improved settlement in the activated sludge. And if we're operating as a BioP plant, then we get better biological phosphorus removal as well. So by just sending the sludge down that route, not only is it practically beneficial because we only have one surplus sludge route, but also we're getting another, say, 10 or 15% more treatment capacity within the existing activated sludge plant. We've also looked at developing standardization for smaller kind of package and mini plants. So package being under a couple of thousand population and mini being maybe 2,000 to 10,000 population, just to try and bring down construction costs. Okay, so I appreciate there's quite a few people on the call who are not from the UK. So I'm just going to mention some of the drivers we've got in the UK at the moment, but I think a lot of these are very similar kind of worldwide in terms of trying to come up with low energy, low carbon treatment solutions and trying to avoid chemicals. But I guess specifically we're seeing in the UK, we're seeing a lot of phosphorus consents. We've also seen trials for total nitrogen. So we're kind of mindful that in the future, we are going to probably have consents for total nitrogen. So we want to be able to remove nitrate if possible and future-proof against those. We also want to try and use as little energy as possible. And we also need to be very mindful of process emissions. So there's a lack of data in general. I think when it comes to nitrous oxide, where things have moved on quite rapidly in the UK, we're a lot more monitoring, and we've seen a lot more global knowledge sharing groups emerging. So it's a rapidly changing place. We've got some examples from the Netherlands that I'll talk about where we've looked at monitoring and mitigating nitrous oxide. And then we're looking at trials in the UK at the moment. Another thing that's featured very heavily in the mainstream press over here is storm spillages. And one of the things we think could be quite an interesting opportunity for granular sludge is how we can use existing assets or build on sites with very small footprint to release assets and space, and how can we most efficiently use those tanks to build up storm capacity. So that's quite an interesting area that we've looked at for a few schemes. And additionally, how we also adapt to changing conditions with more storm tank capacity in the network. How do we then deal with a weaker sewage and a higher frequency of peak flows? So we think granules tend to be pretty resilient to changes in concentration, but also we can look at different buffering options. So when we have our batch process, we would typically have a balanced tank. Unless we have a lot of different reactors. And when we've got a balanced tank, we've got an opportunity as to how we design that for, do we just capture just the peaks and have it as a kind of side stream buffer, or does all the flow go through that? So there's different configurations that we can kind of play with. Okay, so footprint is just some work that was done just to try and compare the footprint of granular sludge to other processes, with it being benchmarked against conventional activated sludge, with that being the largest. And what you can see is we're typically down around the kind of 25% of that in terms of footprint. So smaller than MBBR, smaller than SBR, but comparable in footprint to, say, membrane bioreactors that also tend to have quite a high biomass inventory and no clarifiers. And a good example of this is when we look at this example, Kendal, which is a United Utilities site. You can see the old works on the left, where we've got the whole kind of, it's trickling filter works originally, with those tanks taking up most of the site. And what we've then put in is this Nereda plant. I don't know if you can see my arrow. Hopefully you can. But this box here is just the granular sludge process. And that has then freed up all this other space, which is now a solar farm. But as I was saying, it's a solar farm here, but yeah, it could be additional storm tank capacity. It could release land for other uses. So footprint can play a real key role sometimes. Just another example. This is actually an example from the Netherlands, but I think it just nicely illustrates the comparable footprint. So we've got the original works there in red, treating 140,000 PE. And then the new works, granular sludge process, treating the same flow and load, is much smaller. So yeah, it really shows that kind of around 25% kind of capacity. When we come to energy, a few reasons why the energy tends to be lower. And again, I do appreciate a lot of people on the call may well already understand this very well, but I thought it's worth going through some of the fundamentals of it. But we're using less energy because we don't have any return activated sludge pumping, because we don't have final clarifiers. And also because the granules that I mentioned at the beginning have these different zones in it. So we've got the fully aerobic zone on the outside, and we've got that anoxic zone. We can get really good concurrent nitrification and denitrification through the cycle. And we can control that with the controller. We've got really good kind of control of the DO. So by keeping the DO low, avoiding over aerating, we can make the most of denitrifying, getting a bit of the oxygen and energy back from the nitrate. So that's another mechanism. And also with these batch-wise cycles, we only have to treat until we meet the required effluent quality. We don't have to keep on aerating, as you would do in an activated sludge plant to keep it mixed. So we just treat till it's run the batch till it's treated, and we stop. And there's obviously an energy saving that's associated with that. So as an example, when Southwest Water were kind enough to let us use their site data. This is their data. They had a activated sludge plant side by side with the Nereda Granular Sludge. So they've looked at the energy usage for each. They've standardised it based upon the flow and load that they were treating, and we've come out with this overall average energy saving of about 43%. So yeah, we're really pleased just to see that kind of as a good kind of reference and case study. And we see typically 30 to 50% on most sites, but it's often about capturing the data and kind of proving it that's the key. Just something else interesting about this site, I think it's worth mentioning. I don't know if this is coming to other people's research on this, but there's very high salinity, a lot of seawater getting into the network, and we see the granules being incredibly resilient to it. We don't have any issues at all. And we have done trials in Hong Kong as well, where seawater is often used for toilet flushing. And again, running pilot Nereda there on very high salinity seawater, kind of sewage, no problems at all. So yeah, there's a lot of resilience there we find to kind of toxic shocks or salinity. So okay, if we've got a few minutes left. Yeah, so in terms of retrofitting, just the example on the left I wanted to give here, which is also my backdrop before I started the presentation, is Ringsend at Dublin. So this is a really nice example. It's a double-decker kind of SBR originally. So what we've done is we've retrofitted all of the top deck into granular sludge. We haven't done the bottom deck because, as you can see from the photo bottom left, getting into that to do any construction work would be incredibly difficult. But this is where we've employed a hybrid approach, wasting the granules down into the bottom deck, and it's been very successful at increasing the capacity of the site. And just another example on the right of retrofitting into a conventional activated sludge plant as well. So yeah, I've already kind of touched on these different kind of operating modes. I think one of the things we found for any scheme is really understanding what assets are already on site. Do we treat with or without primary tanks? Do we leave existing primary tanks in? Maybe a mixture partially treated by primary? And how do we look at the overall carbon benefit and balance of that? Do we leave existing activated sludge plant in place? And how do we kind of integrate into it to overall look at each stage? How do we minimise the costs across the whole scheme? So I think a takeaway there is really it's not looking at this as a box in isolation. It's very much how we integrate that into the site assets. Yeah, just another example about resilience. I mentioned about with salinity. This is actually it's a site, a UK site that had a very bad pH shock. So you can see kind of pH 3 coming into the site for a number of hours on that graph at the top, and usually that would kill most activated sludge processes. But what we saw was within 24 hours, the granules had come back to life. And there was a conventional plant on the same site, and that was dead, so completely dead. And so once the granules were back up and running, we could actually pump them across, and then within 48 hours, we brought the other part of the works back into operation again. So it's just a really interesting example, I thought, of just seeing how these granules have that additional resilience. Yeah. And so just a couple more kind of examples, I guess. We're seeing a lot of low phosphorus consents in the UK. We know that it's the same across a lot of Europe as well. So we're seeing typically 1 milligram per litre averages were in the last AMP, and then we're seeing a lot more kind of going down to 0.25, which is consented on 95 percentile. So we have to ensure very low concentration. So typically we know we can meet 1 milligram per litre without chemicals unless it's a very unusual site. But for these lower ones, we would typically always recommend putting in some chemical dosing. And what we've found through a number of trials is that if we dose into the balance tank before the granular sludge process, we need very low chemical doses. So rather than letting the BioP do its thing and then look at tertiary chemical dosing and solids removal, we found if we put it in early on, not only do we get lower solids, but we find that the chemical removal actually benefits the biological phosphorus removal. We both seem to work synergistically. So we can actually, a one-to-one ferric to phosphorus ratio, we're getting down to like less than 0.2. So really, really low. And we've got a number of sites where, because of these trials, they're put in permanent dosing before the plant. And then that means no tertiary treatment. And there's been some pretty big kind of capital savings from that. And just to touch as well, I mean, obviously, if we've got bathing waters, then there may be a requirement for disinfection. And what we found is that generally we get low solids. The effluent quality is good, and at least similar to a comparable long sludge age activated sludge process. But what we find is that really on a site-by-site basis, it's about looking at the UV transmissivity and the dose required, and that would tell you whether or not you need, or whether there's an economic benefit to tertiary treatment to lower the UV dose. So we've got various research on this, and always happy to kind of look for any scheme or what the optimal configuration is. Okay, I think the almost final point was just about the nitrous oxide. So I don't know what experience other people have found in this field, but we did see some negative perceptions early on that because this is a batch process, it's going to be similar to SBRs, but we haven't seen this play out. So we've got some published research here a couple of years ago from the Netherlands where the one site we looked at, it was fairly low loaded, but we came out with a low emission factor of 0.33. And we've done some more recent research at another site where we found that the emissions are a bit higher because it's a higher loaded site, but with various optimizations, we've managed to reduce the nitrous oxide by 50 or 60% against the baseline. And we have found as part of this that different aeration patterns in summer and winter can help as well. So there's quite a lot of functional opportunity to optimize. Okay. So and this is just the example where we started off in summer and we're quite low emissions, then the temperatures dropped. Oh, sorry. Then we saw much more variability. So these dots are the emission factor. We saw it kind of creeping up here. But then what we were able to do was then implement a different process control on the DO, and it's brought back down again to this below 1 emission factor. So simple learning and easy to kind of implement. So yeah, I've already mentioned about kind of plants in operation. Just a couple of quick references just to finish up. And this is Blackburn United Utilities site, consistently meeting a 1 milligram per litre phosphorus consent. And this box here, this is the Nereda plant that's treating the equivalent of what all the rest of the works was doing kind of previously. So yeah, it kept chemical free, meeting fairly low ammonia standards as well. I've already shown you kind of Ring's End. You can see it from a different angle here, but yeah, very, very big site, very site constrained. So you can see why they're keen to retrofit in this scenario. And just finish off. This is the most recent one to commission in the UK, Winchborough for Scotch Water. If you Google Scotch Water Winchborough, then there's some good videos that come up on YouTube and on the Scotch Water website. But this was all 3D designed in advance during the early part of the kind of pandemic. So it's all done remotely. Nearly all off-site manufacture and assembly, and all built out of stainless steel. So a bit of a different construction technique to what we'd normally look at, and that's delivered some fantastic savings in terms of cost and time, and in particular carbon as well. So it's been a real success. So yeah, thank you. See what questions we've got. But as I said, we've got a lot of data. We're always keen to collaborate with academia and others. So any questions after this session, just reach out. Thank you.
Thank you very much for your very interesting presentation. Let me check the chat. I think Sonia, I have you a stable. Okay, there's a question from the Louis biofield. By having stable, longer-lived, and specialized biofilm in the granule, do you see benefits beyond nitrogen and phosphorus? For example, does it help for any of the low concentration, more recalcitrant pollutants too?
Okay, that's a good question. So if I understood that correctly, in terms of, I mean, there was some very, very limited data on things like micropollutants. I don't know if that's quite the question. I think so. Recalcitrant compounds. I mean, obviously, we're running at quite a long sludge age, so I'd say we're comparable to long sludge age activated sludge processes. There's some evidence that certain micropollutants may absorb more into a biofilm because we get a lot of extracellular polysaccharide in the kind of granules. But yeah, I mean, some of your advantages, I guess, are more around things like energy and other things that I talked about. Oh, hands popped up. Question from Charles.
Charles.
Hi, Paul. Thanks for the interesting talk. Obviously, your plants are getting bigger and bigger and bigger. How do you deal with the seeding problem? Because presumably the growth rate of these granules is fairly slow. How much do you put in, and how long does it take to stabilize?
Yeah, it's a good question. I mean, it's getting easier all the time now that we've got more plants. So yeah, I mean, a lot of these big ones, the example there, Ring's End, we brought that on a reactor by reactor basis. So we'd bring in seed from somewhere else, perhaps typically seed at 10 to 15% of the kind of target biomass, and they will grow fairly rapidly. But yeah, I mean, if you're starting a country without any access to granules and it wasn't practical to ship it in, then yeah, you would be looking at more extended commissioning time. But I think most of the recent plants we've done, particularly in the UK, commissioning times up to full flow and load, we're aiming for about 12 weeks typically.
That's faster than I thought it might be. It must create huge difficulties to a brand new country. Is there import difficulties in taking sludge in, etc.?
It's more, I suppose, a logistical thing. I mean, we find that it will stay in stasis for a period of time without degrading. So from a biological point of view, it's perfectly practical. It's more about the actual location of the country. But yeah, I mean, we do find, I mean, it will start to granulate. If we start off with activated sludge, you just need to allow a little more time.
Okay, thanks very much. Good answer.
Thank you, Paul. I have two questions. So I noticed that you mentioned that you can optimize granule size. Can you explain a little bit how can you optimize the granule size and what size is best for performance?
Yeah, it's a really good question. I mean, I want to say, first of all, it's a caveat. I'd say in most situations we don't need to optimize. So it's not that if we don't control and have just the right size granules, we're not achieving consent or anything like that. But what we have found, I guess, is that really, really big granules, there will be less activity in the middle. So we may be holding a little bit of luxury biomass in the process if they're too big. And we also find that I think we're getting better total nitrogen if those granules are just that little bit smaller, but we can control in terms of how we can waste the sludge. We've also got some other research going on in terms of so we're in terms of using coarse bubble aeration just to have kind of more effective kind of cycle times for total N removal as well. So yeah, we've got quite a lot of work happening on an academic side on both nitrous oxide and on total nitrogen.
Okay, so for all of your sites, because we know that for aerobic granular sludge, it is not 100% granule. It is a mixture of the flocks, but dominated by the granular sludge. So from your experience, what is roughly the percentage of the granules in granular sludge?
It's a really good question. I've talked to more of my operational colleagues, I guess, on that. I mean, I wouldn't want to necessarily put a figure on that, and we'd have to go and see what we're actually monitoring. I mean, we do tend to check things like settlement, we check the kind of amount of operational biomass that we have. But I guess the way we design our process, and this is where I suppose the IP sits, is the way we preferentially surplus the non-granular sludge, and that's quite easily controlled. So we surplus off at a certain height, and we take that, and that is generally the weaker or thinner or less granular stuff, and then that goes to a sludge buffer where we thicken it up to maybe 1%, and then that's taken away for sludge processing, and then the water comes back. And because all that's automated, we don't really have to work too hard to control it.
Okay, I think yes, Dionis, do you have any question, please?
Yes, thanks, Leo. Thanks, Paul, for this very interesting talk. I'm Davide Dionisi from University of Aberdeen. Paul, you talked about one of the advantages of the Nereda process over a conventional activated sludge, which is also actually an advantage of SBR processes, is that you said you can aerate only until you need, only for what you need, because which is good, very good point. So you can aerate until the COD is removed. But I'm wondering whether you use in your plants any inline COD sensors or something like that, where you can have live COD measurements and you can control the length of the cycle, the length of the aeration phase based on those readings, or you have a fixed length of the aeration phase. Because if you were able to live measure the COD, you can adjust the timing of the cycles as you need, which would be, I think, a powerful thing to do for your process. Thanks.
Yeah, that's a really, really good question. We tend to use ammonia and redox online. So once we got to a point it's fully nitrified, I mean, typically we would have removed the BOD and the COD as well. I mean, I think online COD instrumentation is sometimes a little more expensive to maintain. So yeah, we tend to find that's more practical, and we tend to also have online orthophosphate. I think one of the challenges we found initially was we were very heavy on instrumentation, and the UK industry is not so keen on having more instrumentation than they need from a cost point of view. So we have tried to rationalise a little bit depending how stringent the consent is, but we do, as a minimum, tend to like having ammonia and redox and pH on each reactor, and then that gives us that kind of control.
Thanks.
Okay, thank you so much, Paul. I think there's a still question in the chat, but because of the time concern, so you can answer the question probably in the chat.
Sure, yeah, I'll pick them up in the chat. Thank you.
Yeah, I will, yes, invite the second speaker to give us the presentation. Our second speaker is Dr. Xiaoyuan Zhang, who is the Associate Professor of Environmental Engineering at Nankai University, China. Dr. Zhang has extensive experience in low-carbon integrated process for energy and resource recovery from wastewater. She also specializes in the development of functional materials and engineering process for the decontamination of radioactive water. With over 50 journal papers published, her research has contributed significantly to both environmental engineering and sustainable water management. Dr. Zhang is currently collaborating with the University of Southampton on ammonia recovery project funded by the Royal Society. Today, her presentation topic is Algal-bacterial granular sludge for low-carbon wastewater treatment. Dr. Zhang, please.
Okay, thank you. Okay, good afternoon, colleagues and friends. I'm Xiaoyuan Zhang from Nankai University, China. It's my great honor to be here to present the topic Algal-bacterial granular sludge for low-carbon wastewater treatment for the Aerobic granule workshop. Thank you for the excellent speeches given by Prof. Paul. And okay, next slide. Okay, I will do this presentation based on the following five points. Let me start from the background challenges with current biological treatment processes. Okay, next. Okay. Current biological treatment processes in wastewater treatment plants are still predominantly biological oxidation-based design, result in high energy consumption, low resource recovery, large production of waste sludge, and greenhouse footprint. Roughly 50% of the organic carbon in wastewater is emitted as carbon dioxide, while the residue organic carbon was accumulated in sludge, and only 30 to 40% sludge in the carbon in the sludge could be converted into biogas through anaerobic digestion if we use the anaerobic digestion. This means approximately 50% of the potential energy in wastewater is lost, leaving an overall energy efficiency is only 20 to 50%. Okay, next. However, existing BNR have common drawbacks, as we all know that the most of the nitrogen gas the nitrogen are from the higher high energy extensive harbor bulge erection, and so the photo fixation of nitrogen and then go to the food chain and go through the nitrogen transformation and finally as the ammonia in the wastewater. So all the existing biological nitrogen removal processes such as the nitrification, denatrification, denatration, and even though the NMOX processes have other common drawbacks. For example, the need for external aeration for example for the nitrification, denatrification, and the reliance on organic matter for denatrification, and the generation of nitrous oxide, which is a potential high greenhouse gas with around 300-fold compared to carbon dioxide. Just like the first speech by Prof. Paul, that we should pay more attention to N2O because even all biological wastewater treatment technologies will generate N2O, and the large variability in reported N2O emission for classical wastewater treatment technologies. And the last one is the BNR processes have no ammonia recovery, so it will cause the contribution to significant carbon emissions. So these factors make biological nitrogen removal an unavoidable carbon source in municipal wastewater treatment. Okay, next. This is the second part for the Algal-bacterial granular sludge. Namely, the abbreviation is the ABGS. ABGS, just as the granular sludge, is part of the granular sludge family, alongside the anaerobic and aerobic granular sludge. As we can see from this image, the 2D macroecology of ABGS enables highly efficient reactions within millimeter scale distances and highly efficient utilization of electron donors and receptors, making the process energy efficient, and also it has a small footprint. Thus, the algal-bacterial granular sludge process is a green and sustainable solution for low carbon consumption wastewater treatment. Okay, next. Microalgae can absorb 10 to 50 times more carbon dioxide than terrestrial plants, providing a promising solution for mitigating climate change. However, maintaining algae growth requires continuous external nutrients such as ammonium and phosphorus. We have a rough calibration. For example, for every 100 kg of carbon dioxide fixed, microalgae require 4.9 kg of ammonium nitrogen and 0.7 kg of phosphorus. Okay, then the question then becomes, where can we get the source for these nutrients? Okay, next. In fact, municipal wastewater contains approximately 40 microgram per liter of ammonium nitrogen and 5 to 10 microgram per liter of phosphorus. Also, it has a constant supply of fresh water, making it an ideal nutrient source for microalgae. If we see from another side, actually, globally, municipal wastewater accounts for around 380 billion cubic meters per year, providing 15.2 billion kg of ammonium nitrogen and 2.85 billion kg of phosphorus. It's just a rough calibration, but we can see from these figures the nutrients such as the ammonium, nitrogen, and the phosphorus amount is very huge. Okay, next. Building on this, just considering the nutrients in the municipal wastewater, and we need to use the biological treatment processes to treat the municipal wastewater. So we conducted a series of investigations to assess the feasibility and efficiency of microalgae and bacterial granular sludge. I will use ABGS in my following presentation to treat the municipal wastewater. Focusing on its nutrient removal performance and the effluent quality. As we can see from this image on the left side in these slides, we found that ABGS can achieve over 85% removal of organic matter, nitrogen, and phosphorus within 6 hours. We use the municipal wastewater we use is the synthetic wastewater, and we can see this removal rate is much faster than conventional wastewater treatment methods. Furthermore, we found that the nitrate and the nitrite concentrations were as low as 0.84 microgram per liter and 0.4 microgram per liter respectively, and the effluent turbidity is in the range of below 1 NTU. And these results clearly show the potential of ABGS process for highly efficient municipal wastewater treatment. Okay, next. Okay. as I just said in the last slides, is the performance of the ABGS process, and we further look into the microbial community in ABGS. The structure and functional analysis of the ABGS microbiome identified was we can see from this chart. It is most of these species as the dominant species. And the species in the outer layer of the granule primarily drives photosynthesis, while aerobic heterotrophic bacteria in the same layer generates carbon dioxide to support algae growth. As such, most COD, nitrogen, and phosphorus were utilized by different heterotrophic microorganisms in the granule, while generating carbon dioxide for microalgae. Also, the metagenomic analysis revealed higher abundance of ammonia transport-related genes and lower abundance of ammonia oxidation-related genes. That's the reason why we found the nitrate and ananitrate could unlikely be produced because the very low abundance of ammonium oxidation bacteria and nitrification related genes. Okay, next.
Dr. Zhou, just for the time concern, can we just go quickly without too many detailed data description?
Okay, another stoichiometric analysis revealed that bacterial elimination removes about 49% of the total organic carbon, while microalgae and bacterial removed 59% and 25% of the nitrogen respectively, result in an overall nitrogen removal efficiency of 84%. And next. Okay, I will be quick. In addition to the targeting conventional pollutants, we also explored the potential of the ABGS for the treatment of emerging contaminants, and we also further expanding its application in wastewater treatment. And we found that ABGS demonstrated strong tolerance to emerging contaminants, like the tetracycline and cadmium and zinc oxide nanoparticles. Next. Okay. This slide shows the structure and the functional analysis of microbiome. I just show the because I cannot show my. Okay, I just show the right figure that we can see the main shows the defense mechanism ABGS to stresses, and other details can be found in our published papers, and I will not show more details. And next one. Moreover, we have done some experiences to find if we all know that microalgae can capture carbon dioxide, and we want to see the effect of carbon dioxide. The effect of carbon dioxide for the ABGS processes, and our results shows that the ABGS can capture carbon dioxide for carbon negative municipal wastewater treatment, and the carbon dioxide could promote the growth of algae and the phosphorus accumulating organisms, and maintain the compact structure of ABGS and enhance the organics and phosphorus removal of ABGS process. That's due to all the reasons that carbon negative operation of ABGS could be achievable. So the capture of carbon dioxide and other industrial origin. Okay, next. Okay, this slide shows the conceptual processes combining anaerobic membrane bioreactor with microalgae culture. Because we all know that there is COD, nitrogen, and phosphorus in the municipal wastewater, and through the anaerobic membrane bioreactor, the soluble COD can be mostly removed by the anaerobic membrane bioreactor, and the residue nutrients in wastewater can be used by the microalgae, and the carbon dioxide produced can be produced from the co-located incineration plants of municipal solid waste, can create the origin of carbon dioxide. So this process can create a situation of one stone killing two birds. And if the and we have do some calibration. If the proposed process were adopted to treat the municipal wastewater worldwide, and the total carbon dioxide capture potential would reach to the 398 million tons. And also the soluble organic matter in municipal wastewater can be directly converted to biomethane through the anaerobic biofilm, and the recovered energy from municipal wastewater could significantly offset the operation-related energy demand. Okay, next one. So considerable efforts have devoted to exploring possible ways to recover the resources from the various forms of microalgae biomass, and the ABGS process can offer a promising future for wastewater treatment, combining the economic and the environmental benefits. And all the biomass can be used for the used to the valuable projects such as the biodiesel, biomethane, lipid, and protein. Okay, next. Okay, so next. And this slide shows the two traditional pathways for microalgae resource utilization. As I just talked earlier, that we can use the Algal-bacterial granules / biomass. So how can we use it? We can use, and this figure shows the two pathways. One is the anaerobic digestion, and one is used directly to the as the biofertilizer. The resource utilization path of microalgae largely determines the carbon sequestration potential, and we have done some roughly calibration, and we can see from this data that the microalgae bacterial granule process eventually becomes a carbon source instead or carbon sink. So we need to find some new pathways for the resource recovery from the Algal-bacterial granule sludge biomass. And also it's a great challenges for this process. And next, and so as I just talked earlier, and I do some quick comparison of the conventional biological processes and the ABGS, and we can see that the ABGS processes has the advantages of low aeration requirements, very low NO2 emissions, as most of the nitrogen was assimilated in the biomass, not also the nitrification denitrification process, and the nitrogen, and it can bring us the nitrogen recovery and capture the carbon dioxide. So these advantages makes the ABGS process more environmentally friendly. Okay, next. So we just can get the all the benefits that ABGS process can be regarded as a water, energy, resource, and a carbon neutral factory. Okay, next. Okay, next. So due to the time limit, and we just go to the summary of ABG advantage. Firstly, ABGS achieves a self-sustainable balance of oxygen and carbon dioxide between microalgae and bacteria. Second, ABGS can effectively remove regular pollutants and emerging contaminants from wastewater. And the third, ABGS can use natural light to fix carbon dioxide and wastewater nutrients into biomass, which can then be further used to produce valuable products. And this is a very long way, but a very promising way for us to explore. And finally, ABGS may open a new window for carbon neutral wastewater treatment with waste recovery. Okay, that's all I share with you. Thank you. Okay.
Thank you very much. Thank you very much for your interesting presentation. I think probably, Luis, we can give the 5 minutes for QA.
Absolutely, yes.
Yeah. So I think Sonia has a question to you. Very interesting on CO2 uptake. Is there any publication on this? Yes, I believe there's a lot of publication. Interesting to know how CO2 from industrial waste gas can be ended and what problems impurities cause, or is it a good way? Sorry. Oh yeah, is it a good way to clean gases without adverse effect or granules?
Okay. Okay. So can I answer it now? Okay. The first question actually is a very good question. And I just show the conceptual process which can combine anaerobic membrane bioreactor with microalgae culture, and then the carbon dioxide used was from the co-located incineration plant. Actually, there is with the background in Singapore, because the incineration plants and the municipal wastewater treatment was built together. So it's a good example for us to follow, that maybe we can use the benefit of the co-location and then use the waste carbon dioxide and transfer the carbon dioxide to municipal wastewater treatment plant. So it's the background from the co-location case of Singapore. So we get the conceptual process. Okay. This is
okay. Yes, thank you. Thank you for your very comprehensive answer. I think another question is from Sonia about the light requirement at large scale.
Yeah. Actually, we have also paid attention to this one, because we all know that if we want to use the Algal-bacterial granule sludge process, the light requirement is important. And until now, that we have tried many, I have do many trials for the light usage in, for example, the sunlight. Actually, the light essentially of sunlight can directly be used in our experiments, but there is a problem is not about the light source, but about how can we make the light penetrate into the waters and be used by the biomass. And I don't know and I also can recommend you a paper that we have done some many researchers related to different light sources and different light intensities, and also the stimulated light source and the actual light source. Okay, and I can recommend a paper. Okay.
Yes, that's good. So because for wastewater treatment plant, so that means and actually the reactor design could be quite different. I think that Louis also asked the question about how does the process cope with fluctuating light levels over the year? Is this a solution for sunny climates only?
Yeah, because we all know that the light, even we have different weathers, and some days are sunny and some days is cloudy, and maybe the light source is not consistent. But I think if in the practical application for this process, we can use the sunlight in the sunny day and use the use the stimulated lights, for example, the LED lights, for the process. And actually, the LED is not. It's very energy efficient and can be used in the treatment plant. Okay, thank you.
I think I have the question, and I'm very interested in the answer, because this is the cultivation of bacteria and microalgae. And you just now show the slides with 48 hours, different batches of the CO2 removal, nitrogen, phosphorus removal. But how about the long-term operation? How about the stability of the bacterial portion and the microalgae portion? Is there any change or variation of the ratio between the bacteria and microalgae? Especially, yes, in the lab, as well as probably in the site. So probably you can tell us in the lab if there's any variation over the time in terms of the bacteria to microalgae ratio.
Yeah. Okay, that's a very good question. As we all know, that the microbes can be cannot keep all consistent all the way. Just if we change the experiment conditions, but in our laboratory experience, the microalgae and ratio and the bacterial ratio can be consistent if we keep the effluent characters be consistent, and after long operation, the microalgae and bacterial ratios will keep consistent. Also, as you said, that the species may become different because some granules will become bigger and the species of the algae will become different. If it will have these questions, but we all know that the microbes can have their own adaptation to the environment conditions, and they will change their performance. They will change the ratio in the granules to use the pollutants in the wastewater. So in our laboratory work, we found that the effluent quality can be kept consistent, but the species in the ABGS can become different. Okay, thank you.
Okay, actually, we have a lot of questions, but because of the time's concern, we can't keep you for too long. So thank you so much for your interesting presentation. Also, many thanks to Paul Lavender, give us both very insightful and interesting presentation. I think we can end now, Louise.
Oh, yes, and thank you to everyone for coming over lunchtime. We do have 2 more webinars, Tuesday and Wednesday. All of the details are online, so do join us again.